Hello, my name is Asger Gjusgaard Jørgensen, and I am a co-founder of Aptashape. First, I will thank you very much for allowing us to present our technology and show what we can offer to industrial and academic partners. We are excited to introduce Aptashape, a technology that turns the molecular signature of proteins and biofluids into a digital fingerprint. Using billions of specialized chemical antibodies called aptamers, Aptashape can detect disease-associated protein signatures long before symptoms appear. This makes Aptashape a powerful tool for early diagnosis, prognosis, disease monitoring, and for monitoring of treatment responses. In the next slide, we will explain how Aptashape works and explore how you can apply it in your projects. So, the Aptashape technology is about identifying complex patterns in biofluids, and we focus on identifying changes in the protein composition in relation to diseases. The Aptashape technology is an aptamer-based technique. The first step in the Aptashape platform is to develop a comprehensive aptamer library able to recognize protein changes correlating with the condition of interest. We call this step for aptamer panel training. An example could be to develop an aptamer panel able to identify early diseases, and we therefore expose a random aptamer library to patient samples representing the condition. The next step is to identify aptamer profiles able to distinguish between different conditions. For example, healthy controls, early disease, and late-stage disease. We do this by exposing our trained aptamer library to multiple samples representing these conditions, leading to identification of sample-specific fingerprints. These patterns, found using next-generation sequencing and machine learning, help us identify aptamers capable of describing the sample characteristics. This allows us to develop fast, cheap, and high-throughput tests based on specific aptamer biosensors. Additionally, the aptamer biosensors can be used to affinity purify the target proteins under native conditions. It allows us to identify and evaluate candidate biomarkers that provide novel insights into disease mechanisms. So, before I present some application of the Aptashape technology, let's highlight some of the key advantages. Aptashape measures proteins, providing a picture of the current situation and unbiased screening of protein biomarkers. Using machine learning, it identifies the best aptamer sensors to answer clinical questions. The aptamers can directly be used to measure protein biomarkers. The technology relies on synthetic reagents and cost-effective DNA sequencing. The analysis takes about 1 day, and the platform is designed for high throughput, enabling automation. I will now introduce what we do at Aptashape. We focus on 3 main categories. The first, diagnostic and prognostics. The second one, monitoring of treatment responses. And the third, stratification of patients. In the next slides, I will share some of our proof of concept studies to show how Aptashape technology can be used. In our first publication on the Aptashape technology, our goal was to identify aptamer biosensor profiles that distinguish between healthy individuals, early-stage bladder cancer, and late-stage bladder cancer. We identified 3 different panels of aptamers in the test cohort and validated the performance in a new validation cohort. first panel consisted of bladder cancer-specific aptamers that clearly separated bladder cancer and healthy controls. The second panel was bladder cancer-specific aptamers, distinguishing between early and late-stage bladder cancer. The final panel combined all aptamers, separating healthy and cancer samples on the first principal component and cancer stages on the second principal component. We then performed homology analysis on the aptamers and identified aptamer families based on conserved sequence motifs. Using these aptamer families, we conducted affinity purification of plasma proteins and identified key proteins, including C4b binding protein, complement C3, and complement factor H. This is an example of how AptoShape can be used in diagnostics. In our most recent publication, our goal was to identify aptamer panels capable of predicting disease severity. We analyzed a test cohort of hospitalized COVID-19 patients and healthy controls and identified aptamers that could separate these two cohorts. We then applied the aptamers upregulated in COVID-19 to a new cohort, including 165 COVID-19 patients. Our hypothesis was that these aptamers could predict COVID-19 severity, and we demonstrated that the aptamers could predict 30-day mortality with an AUC of 0.91 for females and for 0.68 for males. We identified aptamer targets, finding that complement factor H was central to our prediction. Complement factor H and complement factor H-like proteins downregulate the alternative pathway, and complement dysregulation is reported to be central in COVID-19 severity. The last AptoShape project I'll present today was a collaboration between Aarhus University and Novo Nordisk. We investigated how treatment with GLP-1 receptor agonist drugs influenced the plasma protein composition. We used a cohort of black 6 mice fed either a normal chow diet or a Western diet, the latter leading to obesity phenotype. We identified aptamers that could separate these two subgroups. Aptamers capable of recognizing the normal and obesity phenotypes were used to monitor the response to treatment with GLP-1 drugs. Our results showed that mice fed a Western diet and treated with GLP-1 drugs, the blue and green samples in the plot, moved closer to a normal healthy phenotype, demonstrating that AptoShape technology can be applied to monitor metabolic condition in relation to treatment efficiency and disease monitoring. The AptoShape technology can be used to investigate different biofluids, particular cell-free fluids. We have already conducted AptoShape experiments on plasma from humans, mice, and pigs. We have also performed experiments on CFS fluid from human. We have performed experiments using milk from humans and bovine. We have performed experiments on beers and urine. Today we focused the presentation on our studies with plasma, but we are also excited to discuss other sample types with you. I will end this presentation by inviting you to contact us for further discussion about project opportunities and what AptoShape can bring to your project. We are very interested in industrial and academic partners who have an interest in unbiased profiling of biofluids, monitoring and stratification of patients, are searching for novel biomarkers, and we have experience with metabolic, cardiovascular, neuronal, and infectious diseases. Thank you very much for your attention. We welcome for further discussion and encourage you to reach out to us. Thank you.